In medical science, especially if you want to translate from the technical lab to the clinical setting, the tissue context and the connection between things are really important. The mission of my lab is imaging across scales. We try to live this principle by providing not only the microscopes but entire imaging pipelines that allow to go all the way from tailored image acquisition to perfect fitting, data processing and image analysis up to the full interpretation of the data sets we have. The Rudolf Virchow Center for Integrative and Translational Bioimaging is our home and it's our base here, and that's very important for us to be at the clinical campus with this technology-driven institute. My laboratory is specialized in cardiovascular disease and neurological problems or disease-related imaging. In cardiovascular whole organ imaging, the challenge is to do high-resolution imaging so that we can see single cells and the full context of the vessel tree, for example, in the organ. Therefore, we have to make the organ transparent. We have to use a high-resolution light sheet fluorescence microscope, and we have to deal with a lot of data, large data sets for our imaging pipelines.
We collaborate with the Heinze Lab on almost every project we are doing, and we've been doing this for more than 15 years now. And the reason is that we are embedded in an environment here in the Rudolf Virchow Center which allows us on a short note to discuss scientific problems and to find solutions. And for platelet researchers, high-end imaging and advanced imaging methods are absolutely instrumental, because platelets have a number of features that require advanced technology. They are really small, with 1 to 2 microns in diameter. They are highly reactive, and they operate under flowing conditions. And all this requires high spatial and temporal resolution in order to see platelet function at the molecular and at the cellular and finally at the organismic level.
For us, it's important to understand when and where platelets do interact with other immune cells or the vessel wall. And for this, you need to have a resolution which you cannot get with MR imaging. So we need fluorescence microscopy, and we need advanced fluorescence microscopy to be able to penetrate deep enough into the tissue. And only with the help of these tools we are able to understand the cellular interactions of disease progression. Our collaboration with the Heinze Lab is critical for our research. It's, I think, a very complementary mindset and interaction between the two labs. And we have regular meetings where we actually come with our wish list, so we'd like to have this and that. And they then tell us what's realistic, what needs some effort, and what's completely out of scope.
We are collaborating with the Heinze Lab in a project where we want to detect subtle changes in nerve fibers of patients with different kinds of peripheral neuropathies. With a conventional microscopy, we quickly reach our limits. So in the Department of Neurology, we collect skin biopsies that can easily be obtained from patients, and then use super resolution microscopy to identify subtle changes in these nerve fibers. That can then be a diagnostic marker in the future and will also help us to identify the early changes in nerves to find targeted treatments in the future.
The super resolution microscopy allows to see things that the conventional light microscopy cannot. The biggest challenge to get the image is to correct for optical aberration, because these tissues, they distort the light when you go through, to stain specific regions of these tissues, and the contrast in these tissues are really challenging as well for super resolution microscopy.
A big part of my work is to translate between different disciplines, because biologists, engineers and physicists often speak very different languages. Imaging workflows and data analysis strategies are just as important as the microscopes themselves. So good workflows transform complex visual information into meaningful numbers that allow us to compare conditions and draw biological conclusions.
challenges for the big amount of imaging data we produce today is, on the one hand, very obvious. We need the infrastructure, computing facilities, cloud computing, including storage, high-performance computing, and also automated pipelines. But on the other hand, we also want to communicate a lab culture for open science and good scientific practice, so attached to modern, fair principles of research data exchange.
Over the last decades, we have put a lot of effort in developing new machines, crazy super resolution techniques. We have put a lot of effort in programming scripts, artificial intelligence pipelines, and so on. But what we really need is to bring these people together and to train the new generation of scientists so that they understand the importance of these connections, to push this further and close the gap between the disciplines further.